Because DNA contains genes that encode proteins, let's consider an example organism like a worm. The worm is diploid. It has two copies, or homologs, of each of 3 chromosomes in this example. Each horizontal line represents a DNA double helix, a chromosome. So each line represents the DNA sequence of the chromosome. The chromosomes are numbered from largest to smallest. Say genes F, G, and H are on chromosomes 1, 2, and 3 at the locations indicated. These gray alleles encode gray proteins, and genes F and H have shapes that fit together to produce ATP, for example. Different individuals can have different DNA sequences, and those differences are caused by random mutations that change the DNA sequences of chromosomes. After mutation, the distinct versions of genes, called alleles, can produce structurally different versions of the same protein. Here, mutations in genes will be indicated by changing the section of the chromosome containing that gene to a new color. From this genotype, here is the set of proteins this worm would produce. Despite mutation and a change in the protein structure, the proteins can still work together to produce ATP. So this organism is still healthy. If instead we found an individual with this genotype, 2 red F and 2 blue H, then we would imagine that they would not be healthy. They might have died at an early age because those protein interactions that are essential to produce cellular energy cannot occur. In some combinations of mutations can be good, and some can be bad. These alleles that don't function properly together, like the blue version of protein H and the red version of protein F, define a genetic incompatibility. If those alleles are in the same cell, in the same organism, they might not function properly together.